Well, now we're going to move on to, in Bio 121, what is the fundamental unit of all life. And what we're talking about here is what's been constructed through those chemical reactions, from reactants to product to build macromolecules. And those macromolecules are now going to be used to build these single-celled structures, which are called cells, or single-unit structures that are called cells. And there are two major groupings of these. There are eukaryotes and prokaryotes. Eukaryotes meaning they have more membranes inside of them to compartmentalize what actually happens inside of the cells, increasing the efficiency and, of what's actually going on. In this figure here, what I can show you is you have three different populations of cells. Here's looks like tissue. It could be inside a kidney. It's exactly what it looks like, where you actually have multiple populations of cells versus a plant with a cell wall, and then individual sort of prokaryotic structures that live around us. I mean, they completely surround the world that we live in, and even though we have this sort of very human-centric view of the world, anthropomorphized set of rules, we are completely outnumbered by hundreds of trillions of cells in our own body that are living peaceably with us until we do something foolish like try and kill them. But having said all of that, what we need to do is we need to spend some time thinking about how multicellular organisms create a hierarchy. Because if cells are the basic unit, then it means that tissues will be built from cells, and then several different types of tissues can be formed, such that you actually end up having this thing that's called an organ, and then the organs work together to create an organ system. That organ system, when integrated, utilizing signaling systems inside of that multicellular structure through a hierarchy of importance, we then have an entire living organism. So a way to think about this, of course, is to think if here's a cell, it's going to have a series of modifications that are occurring inside of it, and it needs to be built a specific way. It's going to have delimited membrane structures both on the outside and the inside. If there's fluid outside of the cell, there's going to be some form of fluid inside of the cell. And then we start putting them together. They all have to now somehow work together, literally, side by side. Okay, and let's all just assume that these three cells here are the same. Okay? So what Mother Nature is going to do, she's going to figure out a way to integrate this population of cells while simultaneously, let's come over here and make a different population of cells over here. Okay. And the whole point of these really crude cartoons is for you to see that it isn't really so much that we are teaching you the big mechanisms of how this all happens just yet, but that Mother Nature has this ability to create individual units, that basic unit, that will work in cooperation between not only itself, but others that are likely to be more similar to it, and then at the same time be able then, imagine if you will, so that you can begin to sort of tie all of these together. And this is more of a model of how it is you could build something like an organ. And really what I was drawing you here was a population of cells that you find inside of the pancreas, inside of the human body. And the two types of cells I put for you here, you don't need to remember them. These are alpha cells and beta cells, and they're responsible for controlling, literally, the total amount of glucose inside of your body from any moment to any moment. Okay? So if you have a high range of glucose here and a low range of glucose here, Mother Nature needs to try to maintain this range for what you call normal homeostasis. So this is why Mother Nature, okay, creates this hierarchy, if you will, to make sure that things work wonderfully inside your body. It doesn't always work wonderfully, okay? Sometimes things go wrong, but that's the basis of disease, and we're not talking about disease at this point in time. Now, if we think about what else she's doing, how do we actually know this is the case? In lab, I'm going to have you guys go look at a couple of movies about, the different types of microscopes and how it is we actually formed cell theory and why it is we can actually see things with the tools you have, not only in Google, but also in your cell phone and also inside the lab, but also inside specific labs where we have many different types of electron microscopes these days that can actually help resolve things much, 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 much smaller than we ever imagined we could see. how this all works is the difference between magnification and resolving power. Magnification is the number of times you can see things that are smaller than your visual range, and then the clarity is the resolving power. Okay, and that's really what you're working for. Magnification is the process of enlarging an object. That's always easy. Magnifying glass will do that. But a poor quality magnifying glass will not help you understand what it is you're looking at. Now, if you can change the resolution, in this case using under a light microscope versus an electron microscope, you can see a general pattern here, but it requires much more in the way of imagination in terms of figuring out what's going on. But once you've increased that resolution, you can see here the electron microscope gives you this really fine pattern that can be now superimposed on this. Now, the interesting thing here is the process of creating that tissue for the electron microscope changed it from its normal or biologically active state. The light microscope here let it be more close to what you would normally see in the wild. And what you end up having to do is use magnification, resolution, and a fair bit of thinking to actually figure out how all of this works. So from all of this, we actually get what we call cell theory, and the underlying principle for all biology, regardless of the root of biology, anything from cell biology to molecular biology to how this all works, is there. If the cells are the basic living unit, it means all living organisms are made up of cells, and all cells come from pre-existing cells. And that's a really important idea, because when you think about what's actually happening inside the human body, one cell will grow to a specific size, and that cell will then become two smaller cells that should be roughly the same size as where we started here. And this continues, but that doesn't mean that this cell here, this cell here is identical to that cell there. So cell theory, you're going to see as we go along, will have to require that you think about what Mother Nature will ultimately need over here, And if this is you, thinking about here is fertilization, right? If that's you at fertilization, and now you go from one cell to two cells, so this one cell grows, becomes two cells, this ultimately ends up becoming you, you're going to see that even though we have, what happened there, John? Even though we have cell theory sort of describing what's going on, you can actually see that every single cell inside your body is not the same as when it first started. Okay, cool. So where are we going to go with this? Why is it so important? Cells have four common components. There's always going to be some plasma membrane. There's going to be some cytoplasm found inside of it, shown here are these little dots and the things that are inside the solution. DNA, which is the genetic material, is usually found inside a circular, in this case, how do I say this? I want to make sure I say it right for you guys. If we're looking at a eukaryotic cell, and it doesn't appear as though that's what we're doing here, looks like more of a plant cell or a prokaryotic cell. Imagine, if you will, if it's a prokaryotic cell, is a single circular DNA molecule. Eukaryotic cells have a nuclear envelope, increasing the complexity and storing the genetic information inside of that space. It's interesting, they say they're pointing to a ribosome, but they're not actually pointing to anything here. So you should remember that all these little dots, as far as I'm concerned, are the machinery that we're going to use so that you can actually synthesize proteins. And please try to remember proteins inside of cells, definitely inside of this class, are the machines that actually get the work done. And we may not necessarily understand how all of that work will be done, but there will be specific examples as we go through the semester to help you understand that. So here's a generalized structure of a prokaryotic cell, and sort of similar to what we saw in the single structure before. Here we have some modifications for the prokaryotic cells. One, it may have a flagella, it might have these pili. The pili are used for exchanging genetic information. You'll have a capsule of proteoglycan, capsule on the outside. And some of these words may not mean anything to you, but you want to take out a piece of paper, figure out a way to learn what's actually being shown to you here. If you have a capsule on the outside and you have a cell membrane on the inside, That has to mean Mother Nature's invested time and energy in creating something to make sure that the genetic information stays protected here. They're usually much smaller. If you remember that an original picture where I showed you the 3 different sizes of cells, okay, the surface area to volume ratio is more favorable for moving material in and out of a cell, but they also lack the modifications that are required for internal transport. So a way to think about these cells here is that everything that needs to happen in this part of the cell here is happening in this part of the cell. It doesn't matter what's happening over here. Now, why is the surface area to ratio volume such an important concept? And the idea is this. If I magically turned you into a cell, stop and think, look at yourself and go, okay, I am one big cell. You have insufficient membrane surface covering what you call your skin, which would then just be this plasma membrane, to provide gas exchange to allow for there to be life inside of that cell. So when you look at the size of a cell, regardless of the cell type and the modifications that occur inside of it, this is the ideal ratio that will allow for materials to travel into a cell and travel out of a cell. And in doing so, it makes sure that everything that has to occur inside the cell that requires energy can be supported. Okay? And that's a really important concept, because if you think about, say, the rules of 3, right? You can live 3 minutes without air. Okay? If somebody duct tapes your nose and your mouth, you can live for 3 minutes, right? It's going to be painful. It's going to be excruciating. And the more still you remain, the more likely you are to survive that. Cells are exactly the same way. If you make this membrane too big, what happens is it takes longer for things to move in and out to get to where they need to be, and those cells will no longer function. Okay? So these important concepts can now, if we think about like the general structures inside lab, I'm going to have you go find a cell that looks like this. You might even just use this, okay? And you're going to draw it, because you need to know when I'm making a drawing of a cartoon of a cell, because there's no cell in the world that actually looks like this. This is an idealized cell, okay? All of these are going to have specific responsibilities, and you darn right sure I'm going to make sure you understand what a peroxisome is versus, say, a centrosome, okay? So this is a pretty good place to pause here, and then we'll start the next one. But what we're going to do is if this is a cell, and this is a eukaryotic cell, and this is what we spend most of our semester thinking about, eukaryotic cells that are either non-plant or plant-based, we want to make sure we understand how these are built and what they're each doing. So that's where we'll pick up in a minute.